The overall goal of this procedure is to produce slice cultures with clear hippocampal topography and evaluate neurogenesis in the dentate gyrus of the hippocampus. This is accomplished by first culturing hippocampal slices in a culture dish fitted with a special membrane. Next, the dividing cells are labeled with a mitotic marker. The labeled tissue is then cut into sections. Finally, the sections are stained using standard immunohistochemical procedures to identify newborn neurons. Ultimately, immunohistochemistry and cell counting is used to measure cell density and cell properties.
Generally, individuals new to this technique may find it challenging because it requires a lot of practice. Visual demonstration is critical since the dissection steps and the process of transferring tissue from the culture plate to the vibrotome stage can be difficult to learn.
Demonstrating the procedure will be Mosa, a graduate student in our laboratory.
Using a clean scalpel blade, cut the two hemispheres of an isolated rat brain apart. Transfer the left hemisphere back to the large petri dish and place the other PS-side down in ice-cold dissecting solution. View the medial face of the right hemisphere and identify the edge fornix, a prominent band of white matter along the medial edge of the hippocampus. Once identified, cut through the fornix using only about 0.5 centimeters of the scalpel blade. Using two microspatulas, remove the hippocampus by placing the right-hand spatula